The exams are being alphabetized as we speak. I think we should be able to get the keys over to the testing center sometime today. This evening we'll correct the fill in the blanks. I think we'll get the grades back tomorrow. And then by Monday we'll be able to post the grades. It'll be a few more days before we get you'll be able to look at the answers to the exam because we just have, because of the COVID and such, there are some people who are going to be taking a while to get these exams done. So be patient with us as we try to get those exams finished. We should have your grades up, I think, by late tomorrow. So what are we going to do today? Well, we got as far last time as describing the neuron doctrine, and I got a little bit of out of phase in the two sections, so there may be some redundancy again today. But what I want to do is I want to talk about the neuron doctrine and talk about how that was developed, the idea of cellulus and reticulus, and emphasize the concept that the nervous system is constructed of individual units called neurons. And then we're going to talk about these individual units. We're going to talk about the neurons. And we're going to first talk about the structure of individual neurons. And we're going to talk about it in two ways. We're going to describe the neurons first in terms of their structural organization, which is an organization that Cajal could have described in 1900. And we still use this nomenclature, but I want to go through it briefly and talk about that nomenclature, and then extend that nomenclature from what I'm going to call the anatomic or structural description of dendrite, cell body, axon, axon terminal, and Cajal's insight of law of dynamic polarization, moving electrical information from dendrite, cell body, axon to axon terminal, and extend this idea into what I'm going to call the functional description of the neuron. And the functional description of the neuron will have basically the same shape. We're going to change the names a little bit. Rather than a dendrite and cell body, we're going to call it an input region. The input region is going to be characterized by a type of transmembrane protein, a type of channel in the membrane called a ligand-gated channel. It's going to be opened by neurotransmitter. So when you see this term ligand-gated channel, think neurotransmitter-operated channel. And it's going to be operated by a neurotransmitter released from neurons that are right next door. Then there's going to be a conductal region of each of these neurons, and the conductal region is going to be a great long process that's carrying electrical signals over long distances. In the anatomic description, we call it the axon. In the function description, we call it a conductor region because it's conducting electrical signals. And we'll see that the electrical signals that are generated in this part of the neuron are different from the ones in the input region because these signals in the conductor region are being carried out, propagated, initiated by voltage-gated channels. We're going to spend a lot of time talking about ligand-gated channels and voltage-gated channels, but for right now, know that we're going to relegate them to different parts of the neuron. Ligand-gated channels are going to be in the input region, voltage-gated channels are going to be in the conductor region, and then the output region is the region now where we're going to release neurotransmitter, so that neurotransmitter then can communicate to a nearby cell. The output region is going to be characterized by another kind of voltage-gated channel. It's going to be characterized by a calcium channel. And we'll open these voltage-gated calcium channels. We'll use charge to open these channels just like we do in the conductor region, but now, rather than letting in sodium in or potassium out, we'll now be letting calcium come into the cell. The calcium coming into the cell will then trigger vesicles to fuse the plasma membrane and release neurotransmitter by exocytosis. We'll get down into the weeds of all of this. But for now, I just want to think about these two descriptions of the neuron, one being based on its anatomy and one being based on the distribution of its different kinds of transmembrane channels. But one of the characteristics that will come out of the structure of the neuron, whether we're talking about the functional structure or the anatomic structure, is the fact that these are highly asymmetric cells. It's not unusual to have a motor neuron that resides in your spinal cord and goes down to your toe. The neuronal cell body will be confined. It has to be located in the central nervous system, but that axon now, the conductor region, is going to go over a meter. The logistical problem arises because all protein synthesis in neurons occurs in the cell body. And yet, we have to move those proteins from the cell body all on the axon down to the terminal. So this brings up a real logistical problem. We make proteins in one place, we have to deliver them to another place. How do we get this delivery system to work? And we've talked about that some. We've talked about it in terms of transport on microtubules. And in this particular case, we're going to see that proteins are going to be produced in the cell body and move toward the axon terminal. And as they move from the cell body to the axon terminal, this will be called anterograde transport. But there's also transport that moves from the terminal back to the cell body, and this is retrograde transport. Now, anterograde and retrograde transport are all on microtubules. You may recall that the anterograde transport is on microtubules in such a way that it's a plus-end directed motor using kinesin, whereas retrograde transport is also in microtubules, but it's being carried out by the negative end directed motor, dynein. And we'll work our way through that. Now, one of the early sets of experiments in all of this realized that the proteins that were generated in neurons moved at two distinctly different rates. One rate was proteins were moving at about 200 to 400 millimeters a day. And there was another rate where they were moving at 0.5 to 2 millimeters a day. And when they drilled down and began to recognize which proteins were in which group, they found that vesicular proteins were all in the fast exoplasmic group. And the proteins that are moving in the so-called slow component at 0.5 to 2 millimeters a day, these are all cytoplasmic proteins. So again, reminding you that proteins that are produced on free ribosomes are cytoplasmic proteins, and proteins that are produced on endoplasmic reticulum, processed through the Golgi and packaged into vesicles, those are going to be the proteins moving in vesicles and microtubules. And if there's time, I want to tell you about some what I think are really important experiments to distinguish the fast and slow exoplasmic transport. This is where we begin to talk about rabbit retinas. And there's always a question of why are we talking about rabbit retinas. But there's a reason to it, because we know where the cell bodies are, we know where the axon terminals are, and we know how they're laid out in the optic nerve. We can use that anatomy to begin to discern what proteins are moving in each of these components. And finally, one of the points that we'll make, and we may not get to it today, is that slow exoplasmic transport, although it looks far different from fast exoplasmic transport, the slow exoplasmic transport, when it moves, when the cytoplasmic proteins are moving, they're moving at fast rates. But they only move at fast rates intermittently. So it's stop and go, stop and go. And that's the main difference between these forms of transport. The 200 to 400 millimeters per day is continuous. The slow exoplasmic transport is discontinuous. It's intermittent. It's in fits and starts. And we'll talk a little bit about that. But let me finish the story from yesterday of the neuron doctrine. We all know from the neuron doctrine, our description of from yesterday, is that the nervous system is composed of individual units called neurons, and that the neuron is the fundamental structural and functional unit of the nervous system. And this is a concept that arises largely from work done by Santiago Ramon y Cajal. I'll never ask you names, I'll never ask you dates, but I need names and dates to tell the stories. So as I tell the stories, latch onto the concepts. What we see here is that in the late 1800s, early 1900s, there were two schools of thought on the nervous system. One school of thought were said to be the reticularists. And the reticularists held that all neurons are connected to one another by cytoplasmic bridges. That these neurons were all basically one syncytium, one continuous structure of cytoplasm. The other school of thought were the cellularists. And the cellularists said, no, that's not right. The cellularists said that the nervous system is composed of individual cells called neurons. And these two arguments were largely proposed by two leading scientists. One of them was this guy over here called Camillo. Golgi. Camillo Golgi was an outstanding microscopist. He was a pathologist. He worked at Pavia, which was one of the major universities in Europe. And he was instrumental in determining the thinking about the nervous system. And he developed something called the Golgi stain. Now, up to this time, most of histology was carried out using new dyes at the time, in the 1800s, that had been developed out of the coal industry. These were called the aniline dyes, and they were very effective at labeling different tissues. But the problem is when those aniline dyes were applied to the nervous system, there was too much information. You could just see thousands and thousands of cells in every section, and they just couldn't make any sense of it. So Golgi developed this clever stain, and rather than using aniline dyes, the Golgi stain depends on silver and chromium. We now know that the silver and chromium is probably staining the cytoskeleton, but the joy of this particular stain is that it stains neurons in their entirety. Meaning it stains the whole cell. Now, you'd think this would be a problem, because if we were worried about the aniline dyes staining all these cells, this should be even worse. But it turns out, by dumb luck, it only labels about 1 to 2% of neurons. And again, you would think on first blush, if you wanted a specific label, but you only labeled 1 in 100 to 1 in 50 of the neurons, it wouldn't be a very good dye. But it turns out, fortuitously, because it only labeled 1 to 2% of the cells, but it labeled them fully, that this allowed people to begin seeing neurons as the full structure. There was a fellow named Santiago Romanical. And unlike Golgi, who was working pretty much at the heart of the scientific universe, Santiago Romanical was working in Spain. It was pretty much on the outskirts. And he used the Golgi stain. He modified Golgi's technique a little bit, but not very much. And he then applied this stain to the nervous system, just as Golgi had done, but he came up to a very different conclusion. He came to the conclusion that the nervous system is composed of individual units. Called neurons. This story has a kind of, I think, interesting human interest aspect to it, angle to it, in that in 1906, these two individuals shared the Nobel Prize. The Nobel Prize was awarded to Golgi for developing the technique, and it was awarded to Santiago Romanical for using the technique to describe the neuron doctrine. Now, whenever Nobel Prize winners receive the prize, they give a seminar. And you can actually go back and read these seminars. And when Golgi gave his seminar, he got up and gave this talk that basically said, Cajal's got it all wrong. Cajal doesn't know what he's doing with his staining technique. He's a complete idiot. And Cajal got up and was very gracious. He thanked Golgi for developing the stain and providing him with the ability to sort out the problems of the nervous system. To their dying days, they hated each other. Those two talks put these two people at odds for the rest of their days. There's even a political aspect of this. Golgi was very influential both in politics of academics in Italy as well as in politics of the government. And he became very influential, and his theories about the reticular theory were taught well up into the 1930s when everybody else knew they were wrong. But they were taught in the med schools because he was such a powerful individual within those political systems. I find that sort of an interesting sort of caveat on the politics of science. But Cajal had it wrong. Golgi got it, Cajal had it right. Golgi got it wrong. And let me extend Golgi's ideas. Golgi said, try again. Let me extend this to talk about concepts of Cajal. The concept that Cajal came up with, not only the neuron doctrine, but he came up with the concept of the law of dynamic polarization. This is just a fancy terminology. It means unidirectional flow. And much of that insight occurred because he could see the structure of the neuron. And although this is going to be a crude representation of what he showed, he has these beautiful, beautiful drawings. I'll show you one of those in a moment. But this is one of my pineapple neurons. And fundamentally, it says the same thing as Cajal's exquisite drawings show. This spiky region out here is said to be the dendrite. It comes from the Greek dendros, meaning tree, so it looked branched. So there were all these branches of the dendrite. And then he could see from other staining that there was a nucleus in here, so he called this a cell body. And the term he applied to this long extension from the neuronal cell body was the axon. And out here, he called this the axon terminal. He didn't know it at the time, but a few years later, people began to recognize that this axon terminal would now connect to the input region. Let me not start mixing terminology. Connect to the dendrite of the neighboring cell. And this connection between these two individual units was called the synapse. He didn't know that, but what he recognized in one of his main insights of the law of dynamic polarization is that information would flow from the dendrite across the cell body, down the axon, and down to the terminal. It was years before they understood how that information was flowing or being moved. It was a long time before they recognized how the electrical signals. If you put your nose away, I'd appreciate it. Thank you. That this information was flowing in one direction, from the dendrite to the axon terminal, and it was years before they recognized how the information was flowing. It was flowing through the use of ion channels. And what that brings us to is now the functional description of the neuron. So I'm going to draw this neuron the same as I have in the upper panel. And the structure will be the same. We'll see a little later when we talk about sensory neurons that this nomenclature will be invoked. And we'll see that it works just fine on sensory neurons. But I want to use this opportunity to distinguish the early description of the neuron in anatomical terms and begin describing neurons in functional terms. So on this panel, I'm going to draw a neuron, and we're going to label it as the functional description of the neuron. I'll draw it the same way. It's going to be another one of these pineapple neurons. Bless you. But now we're going to change the nomenclature. The nomenclature is going to include, it's going to group together the dendrite and the cell body. This is going to be called the input region. The structure above, which we had named the axon, gets a new name. It's called the conductile region. And down here, the axon terminal is again renamed, and it's called the output region. And as the name implies, as I tried to do earlier, it's going to communicate to the input region of the neighboring cell at a site called a synapse. Well, one of the reasons we have included this nomenclature, we've changed the nomenclature, is that once we began to understand the electrophysiology of these cells, began to recognize that one of the hallmarks of neurons is the different distribution of channel types in the neuron. So that when we're talking about input regions, the input regions were characterized by ligand-gated channels. This is a technical term, and we're going to use it over and over again, but when we talk about ligand-gated channels, what we're really meaning is we're meaning neurotransmitter. Operated channels. And then we find that there are different series of channels here in the conductal region. The conductal region is characterized by a kind of channel called a voltage-gated channel. Another name for voltage-gated channel, this would be a charge-sensitive channel. Where these channels out here in the input region are operated, they're opened, they're regulated by neurotransmitter. These channels along the conductal region are operated by a change in charge. We can draw this in such a way that we can say that there are channels out here that are opened by neurotransmitter. There are a variety of different neurotransmitters. We'll talk about a bunch of them, but they're basically a chemical which will bind to the transmembrane protein, open the channel, and allow sodium to come in. But these particular channels are relegated to the dendrite and cell body, the input region. Then another group of channels, which are only rarely found in the cell body of the dendrite, are now begin to crop up in the cell. These are going to be voltage-gated sodium channels. And their counterpart, which we're going to talk about a lot, are voltage-gated potassium channels. We'll see that working in concert, the voltage-gated sodium channels and the voltage-gated potassium channels are responsible for generating something known as the action potential. And the action potential will propagate for long distances. along the membrane. Of the conductor region. So again, the input region gets its name because it's receiving input in the form of neurotransmitter from neighboring cells. The conductor region gets its name because it is propagating or carrying electrical signals, conducting them over long distances. And those signals, those electrical signals, will arrive at this region here called the output region, where there are another set of channels down here, we're going to talk about in some detail, called voltage-gated calcium channels. The calcium channels, these Ca2 plus channels, allow for calcium to enter the cell from the outside world. And the calcium leads to the opening let me say that differently, leads to the fusion of synaptic vesicles. To cause neurotransmitter release by exocytosis. So we've now come from Cahal's law of dynamic polarization to recognizing that electrical signals are going to start here in the input region, propagate along the membrane down here to the output region, and then at the output region allow for the opening of voltage-gated calcium channels to allow calcium to permit the fusion of vesicles with the plasma membrane, the release of neurotransmitter, and then that neurotransmitter can operate channels in the next cell. We find the nervous system is like a giant daisy chain of neurons connected one to another. So any questions up to there? Yes?
Is it synaptic or synaptic?
Synaptic. These should be. From the term synapse. And we'll talk about synapses a little bit later. Yes?
So the action potentials, is it both the voltage-gated calcium channels and battery channels? Is it both of them?
Both of them are involved. We're going to see, and again, this is a little early for this, but we're going to see the voltage-gated sodium channels are responsible for allowing sodium to come in. And then we're going to use the potassium channels to carry positive charge back out again, so that we get a potential that goes from about minus 70 millivolts up to plus 40 millivolts. Potassium channels open and drive that potential back down to rest again. And we're going to talk in great detail about that. Anything else? Okay, so up to this point, what we've been talking about first is this bizarrely shaped cell, and we've been emphasizing the structural components, and then emphasizing the components within the membrane. Now we're going to change gears a little bit, and we're going to begin talking about what's going on in the cytoplasm. And to describe this, we'll begin with this structure, and I'll just point out that neurons are highly asymmetric. Cells. And we get a sense of that even from my crude drawing, in that this cell body is here and the axon terminal is way out here, a long distance away. It's not unusual in the nervous system to find a neuronal cell body that has a diameter of, 30 to maybe 50 micrometers. Now remember, a micrometer is a millionth of a meter, so this is pretty darn small. And yet, the length of this conductor region, the length of this axon, can be, greater than 1 meter. Even in us, there are motor neurons who have their cell body in your cervical spinal cord or in your lumbar spinal cord, and they will send processes out that innervate muscles down in your hand, or they will innervate muscles in your feet. So that one neuron, a single neuron, whose cell body is located in the spinal cord, will have its output region like a meter away to talk to muscle. If you look at the same kind of neuron in a giraffe, it gets even worse. Because you have a neuron in the cervical region of a giraffe innervating regions of the forelimbs of that giraffe. That's a long, long distance. And yet the rules are the same. There's a neuronal cell body, there's an axon, and most importantly, protein synthesis. Is confined to the cell body. Now the rules here are pretty much the same as we talked about earlier when we talked about cell biology. In the cytoplasm, let me go up to the other one. So I have two drawings. In the cytoplasm out here, there will be free ribosomes. And as we learned in cell biology, these free ribosomes are responsible for making cytoplasmic proteins. But along with those free ribosomes, for generating transmembrane, lysosomal, and secreted proteins. So all the rules we learned earlier in cell biology still apply. We're going to have a Golgi apparatus, that vesicles are going to bud off of the Golgi apparatus. And these vesicles, the vesicles, of course, are going to contain the transmembrane proteins and the secreted proteins and the lysosomal proteins. So the rules are all the same. But we have a real problem in this cell in that the site where we're making the proteins may be a meter away from where we need them. And so that brings up this question of transport. And now when we think about transport, and it's something that's sometimes hard to get your head around, when we think about transport, we're talking about cytoplasm. We're talking about the jelly of cytoplasm inside the membrane. When we're talking about the channels and propagation of electrical signals, that's all going on along the membrane. And just to give you some sense of the sizes here, this membrane is about 6 to 7 nanometers wide. That's a billionth of a meter. The diameter of this cell is not unusual to have it be 10 micrometers. So this thing is 1,000 times bigger than the width of this veneer of the membrane on the outside. This is a giant bit of cytoplasm compared to this tiny bit of membrane. Electrical signals are going to be propagating all over the membrane. But now what we're going to see is the proteins that we generate here, up here in the cell body, need to be propagated, need to be moved, let's not use propagated, need to be transported in the cytoplasm from the axon, from the cell body to the axon terminal. And that's where we're going to head now. So any questions up to there? So let's take the upper panel then and talk about the concept of exoplasmic transport. And again, when we're talking about exoplasmic transport, we're talking about the movement of proteins. And we'll redraw one of these neurons, we'll pretty much keep the same shape, and now begin thinking about what's going on inside the cell, in the cytoplasm. In the cytoplasm, in the axon, called the axoplasm, and think about how we're moving these proteins. And again, much of this is reminiscent of what you learned early on when we talked about cell biology. But let's now put it in context of neurons. So we've established the idea that all proteins are made in the neuronal cell body, our dendrites. And then all along the axon, you'll remember, there were these structures, these tube-like structures, called microtubules. So these straight blue lines are meant to represent the microtubules. And you may recall that in the axon, the microtubules are always oriented in a particular way. And that the negative end was closest to the cell body, and the positive end was pointed away from the cell body. But the rules from above still pertain. That is, we make vesicles. We fill those vesicles with molecules that be secreted. We fill those vesicles with transmembrane proteins, and we put these vesicles now onto the microtubules. And our old friend, kinesin, can now grab at one end to the vesicle and grab with its feet to the microtubule. And because kinesin is a plus end directed motor, it drags this vesicle from the minus end of the microtubule toward the plus end. And these proteins now, these vesicular proteins, are transported by the kinesin at rates of about 200 to 400 millimeters per day. Now, this doesn't sound like a whole lot. I mean, we're talking about, 20 to 40 centimeters, less than a foot. It takes a day to do that. But remember that these guys are being transported by a motor that's making 8 nanometer steps. The feet on these kinesin molecules are moving at an incredibly fast rate, taking these tiny steps, dragging the vesicle from the region of the cell body toward the terminal. And the movement of protein from the cell body toward the terminal gets a name. It's called anterograde transport. Anterograde is moving the cell body out toward the terminal, but there's also another form of transport. And the other form of transport is from the terminal, where vesicles are being taken in by endocytosis. These vesicles then are again put on microtubules. And these microtubules again have their plus end toward the terminal and the minus end toward the cell body. These vesicles now can be mounted on the microtubules through the other motor called dynein. And dynein is now responsible for what's known as retrograde transport. We most often emphasize these ideas of anterograde transport because the proteins are being made here and carried out, but the retrograde transport is really important. One of the things that happens in endocytosis is that growth factors are endocytosed. These growth factors are packaged in the vesicle and they're transported all the way from the terminal back to the cell body. And in the cell body, those growth factors then will affect the genome. Specifically, they suppress apoptotic genes. All of our cells have a series of genes in them known as apoptotic genes. These apoptotic genes are in fact kind of suicide genes. What they do is they organize cell death. And all of our cells have them. In this particular case for the neurons, it's important to turn those apoptotic genes off. And that's what the growth factors are doing. The continuous flow of growth factors from the terminal to the cell body to the nucleus seems to play an important role in keeping these cells alive. Because otherwise, without the growth factors, they induce their apoptotic machinery and they die. So the growth factors and retrograde transport is really important. Both retrograde and anterograde transport move on the fast side. They're moving about 200 to 400 millimeters a day. It doesn't matter whether we're transporting vesicles by kinesin or by dynein. They seem to move about the same rate. But you'll remember that in addition to those proteins that are packaged in vesicles, there are proteins that are in the cytoplasm, things like tubulin and actin. All those cytoplasmic proteins that are generated on free ribosomes. And these free ribosomes generate cytoplasmic proteins. So the cytoplasmic proteins were found early on to move at a very different rate. That they always moved in the anterograde direction. They always moved in the cell body toward the terminal. But the cytoplasmic proteins, these guys were transported at a rate of about 0.5 to maybe 2 millimeters per day. So this was said to be slow axoplasmic transport. So we have slow axoplasmic transport and fast axoplasmic transport moving at different rates. Fast axoplasmic transport is dependent upon vesicles moving along microtubules using molecular motors. But the slow transport was confusing for a long time because what accounted for that difference in rate? What accounted for the rate at which we were moving vesicles and they were moving straight proteins? The way this was begun to be sorted out was a set of experiments that were done in rabbit retinas. You know, before I go on, because I forgot to do this and because I'm now beginning to be out of phase with the two lectures, let me go back and show a slide that I meant to show you before. Sorry about this. This is a picture that I really like, so I'm sorry I didn't show it. It's on the right-hand side, it's showing a picture of Cajal, Santiago Ramon y Cajal, sitting in his laboratory. And he's sitting there in front of his microscope. And behind him are all these different stains. And he's looking very pensively. I'm often thinking that he's thinking that Golgi was a fool. But on the left-hand side are the drawings that he made. This is a particular set of drawings that he made of cells from the pigeon retina, maybe pigeons he was feeding in the afternoon for all I know. And you can see that with the Golgi stain, you see these cells that were stained in their entirety. And there are a couple of classic cells in here. This is a pyramidal cell here. This is neuronal cell body. Here it's dendrites. Here it's axon. And from just looking at the anatomy, from emphasizing the anatomy and studying the anatomy for hours and hours and days, he came to the conclusion of the law of dynamic polarization. The information was moving in one direction. It was moving from the dendrite over the cell body to the axon. He had no idea what the mechanism was. That took another 50 years to sort out. But we now know that's electrical signaling, and that gives rise to the functional description of the axon, or of the neuron. But what we have here is the anatomic description. Again, here's a cell body, here's a dendrite, here's an axon coming out of it. And this was important because it showed the individual nature of neurons. It showed the concept of the dynamic polarization of neurons. And that was the story I was trying to emphasize when we were discussing the anatomic description of the neuron, and then further describing that into the functional description of the neuron and the position of various kinds of channels. But now what we want to do, we do it in the back panel here, is I want to describe a series of experiments, and we'll take this and describe them as experiments to sort out fast and slow transport. Now, these experiments were done in a particular preparation. And they were done in a particular preparation called the rabbit retina. Now, we can't get too much in detail of rabbit retinas, but rabbit retinas are this conglomeration of neurons. But there's one set of neurons that line up, and I'm going to show them as a series of dots in the retina. And these neurons are known as retinal ganglion cells. The point is that the retinal ganglion cells have cell bodies in the retina. And one of the major advantages of the system is the retina is out separate from the brain and is out in a fairly accessible site. So what they could do is they could come in here with a needle and they could inject radioactive amino acids. Well, those radioactive amino acids will be taken up by the ganglion cells. They'll be taken up by all the cells, but they'll be taken up by the ganglion cells and converted into proteins. Now, the major advantages of this preparation is that these ganglion cells have their axons that all gather together. So these are axons, or conductal regions, in the optic nerve. These axons, I won't draw all of them here, but these axons have output regions in a region of the brain called the lateral geniculate. The anatomic terms are not important here, but what's important is that they knew that they could get protein synthesis at this site. And they knew they had great long axons that they could now exploit. Because what they could do is they could inject radioactive amino acids in here, and then they could wait for a period of time. They could kill the animal, dissect out the optic nerve, and then begin taking sections of the optic nerve. And by waiting different periods of time, they could sort out and write a graph, which was time versus radioactive proteins. And when they did that, they found that there were two components. One component was moving at about 200 to 400 millimeters per day. And these were all vesicular proteins. And the other component were cytoplasmic proteins. And they were moving at about 0.5 to 2 millimeters per day. And that was the first indication that we had different kinds of proteins moving in the different components. And then it took about 10 years to sort out this component and another 20 years to sort out this component. And I'll finish the story tomorrow and tell you the story about how these proteins are moved in the cytoplasm.